Good morning. This lecture video is going to go over the cell cycle and how that cell cycle is regulated. The cell cycle is a sequence of events where the cell alternates between periods of growth and then periods of active division in order to make a copy of itself. There are essentially 4 stages, with the M stage or the mitosis stage being further broke down into other or sub-stages. The end result of a cell completing a cell cycle is going to be the production of a new identical cell. So when this process is over, we will have started with one cell, and then we will end up with 2 cells that are genetically identical. There are 2 main parts of the cell cycle: interphase, which includes G1, S, and G2, and then mitosis, which is going to include prophase, metaphase, anaphase, telophase, and cytokinesis. Starting with gap 1 or G1 stage of interphase, the cell spends the vast majority of its life cycle in interphase in one of these 3 stages, G1, S, or G2. During the gap 1 stage, or the G1 stage, this is going to be where the cell is basically chilling out, carrying out its everyday functions, all the things that it needs to do just to survive. Okay, it's growing, it's growing quickly, it's carrying out its normal routine, it's just, doing its job, doing its business. Nothing super eventful is happening, kind of like a maintenance type of stage. As we enter into the S phase, what's going to happen is that the DNA is actually going to make a copy of itself. So remember, when we want to make a new cell, we want it to have all of the genetic instructions in it that the old cell had in it, right? We don't want to have faulty genetic information. We need to figure out a way for us to have genetically identical cells. So during this S phase, the DNA is actually going to make a copy of itself so that one copy can go into the new cell and the other copy can kind of stay in the original cell. This is where we actually start to see chromosomes forming during this stage. You can actually see those structures under the microscope because the DNA condenses really, really tightly, so you can actually see these X-shaped structures, which we refer to as chromosomes, which are just wound up pieces of DNA. Then we transition into the G2 phase. During the G2 phase, preparations are made for the cell to begin to actively divide into 2 cells. All of the organelles will make copies of itself. The cell will carry out some routine checkpoints to make sure that everything is ready to go before it enters into the division stage, which is the M phase. So G1, S, and G2 are going to be preparatory stages, and that's why we call them interphase. And again, the cell is going to spend the vast majority of their time in those stages. Mitosis is a stage of nuclear division. So as we go from G1 into the first stage of mitosis, which is prophase, the end result of mitosis is going to be 2 identical daughter cells. 5 stages: prophase, metaphase, anaphase, telophase, and cytokinesis, and we're going to go over those real quick. During prophase, the chromosomes become visible because they're squinching together so tightly. The nucleolus of the nucleus disappears, and the nuclear membrane actually dissolves. So there's no more like compact nucleus. Everything, all this genetic material is floating all over the cell. Spindle fibers also start to form, which are going to be fibers that are kind of going to, think of them almost like chromosome leashes. They're going to direct the chromosomes where to go as it continues through this mitosis process. The next phase is metaphase. Easy to remember, M for middle. The chromosomes, but the spindle fibers are going to line the chromosomes up along the middle of the cell. So we get this line of chromosomes. That takes us to anaphase. A for anaphase, A for apart. The spindle fibers will then shrink and start pulling the chromosomes apart into opposite poles of the cell. So again, this copy of the chromosomes coming to this side, this copy of the chromosomes going to this side. During telophase, the nuclear membrane is going to start to reform around those chromosomes on each end of the poles. The nucleus starts to reform, the nuclear membrane starts to reform, and we basically are going to have, at the very, very end of telophase, one cell that has two nuclei in it. And the last phase then is going to be when you can see how it's starting here. The cell's going to start to cleave in the middle in order to start to separate into two cells. And this process is called cytokinesis. Some scientists say cytokinesis is its own separate category. Some of it say that it's part of telophase. Ultimately, it doesn't matter, but this is the stage where it's actually going to end up forming two new cells that are genetically identical. Each one has a copy of the DNA that was copied during the S phase. If you were to look at a cell, here's an onion root cell under the microscope. So like you can see some of these stages, these ones that have the fried egg look, those are going to be in interphase. When you start to see some of these speckles start to form, that's when the chromosomes are condensing. So these would be prophase. This is a good one here, prophase. You can see here anaphase, where the chromosomes are pulling apart to the opposite ends of the cell. Here's another anaphase. This one's actually probably a little bit better. So you can see how they're migrating to opposite ends of the cell. Again, here's an interphase. Here's a prophase. You can start to see the lines a little bit, the chromosomes. Here's metaphase, where they're all along the middle. Anaphase, where they're pulling apart. And then telophase, where you're going to end up with two separate cells. How is this cycle regulated? This is really important because cancer is a disease of the cell cycle. This process, something goes very, very wrong with this process. And most of you know someone who has had cancer and can know how devastating it can be. In 1980, scientists discovered what actually tells a cell when it should divide or duplicate their chromosomes, or in other words, when it should go from one stage of the cell cycle to the next stage. This was a huge breakthrough because, again, cancer causes uncontrolled growth and division of cells. So finding out what regulates this cell process has been a very, very huge, or made a huge impact on our study of cancer and trying to figure out a way to cure it or stop it. Cyclins are the name of the proteins that regulate the timing of the cell cycle. And we put cyclins into two different categories, internal regulators and external regulators. Internal regulators are going to be proteins that respond to events inside the cell. They're going to tell the cell when to go from one stage to the next stage. So going from G1 to S, or going from S to G2. And they're going to monitor what's happening inside of the cell. So for instance, when you go from the S phase to G2, these internal regulators are going to basically like proofread that DNA, make sure everything looks good before it will tell the cell that it can go into that G2 stage. External regulators are going to be proteins that respond to events outside of the cell. So they're going to tell the cell when they need to speed up or slow down going through this process. So for instance, if I injure myself and I need to be making new cells to replace my injured cells, external regulators actually tell my cells to start going through the cell cycle at a much faster rate in order to replace the injured cells. And then again, once that gap between my wound starts to fill in with cells, it will recognize that and it'll tell the cells to start slowing down so that we don't end up with this huge overgrowth.